Everyone, this is Farha of Group 316. This is my second video and the topic is antibody and antigens. A simple definition of antibody. It is a Y-shaped protein produced by our immune cells called B cells to attack and fight with foreign bodies. Antigen. It is a foreign substance that enters our body and triggers immune system to respond against it. For example, bacteria, viruses, fungi, allergens, venom, and other toxins are the antigens. Types of antibodies. Antibodies are also called immunoglobulins. There are 5 main classes of immunoglobulins. The first one is IgG, immunoglobulin G. It is the most abundant antibody, 65% to 70% found in blood plasma. Plasma cells secretes IgG antibodies. The structure is monomer and single Y-shaped unit, as you can see in this picture. It has the 2 antigen binding sites, a hing region, light chain, disulfide bridges, and the heavy chain. IgG produce in both primary and secondary immune response, but more abundant in secondary immune response. IgG provides long-term protection and cross the placenta to protect fetus. How does IgG antibody works? Example, it is a bacteria and it is an antigen. IgG antibody binds and activate complement protein system and formation of MAC, membrane attack complex, and enhance opsonization. Here it is a virus. It has the antigens on its outer layer and IgG antibody binds to it and neutralize it. It is called neutralization. Here the free antigen is roaming, then the IgG antibody binds and precipitate antigen phagocytosis initiation occur, process called precipitation. And here the IgG antibody crosses the placenta and goes into the fetus and it gives the passive immunity before birth. The second is IgA immunoglobulin A. Structure monomer or dimer, 2 Y-shaped units linked, secreted by plasma cells in the form of dimer, found in mucosal surfaces, respiratory, gastrointestinal, genitourinary tracts, saliva, tears, breast milk, mother to baby. IgA passes to baby. It has the 2 Y-shaped units linked together by J chain. Functions: protects mucosal surfaces from pathogens. It is a dimer of IgA. It can bind to 2 antigens of the 2 bacteria at a time. Third one, IgM, immunoglobulin M. The structure is pentamer. 5 Y-shaped units are linked together by disulfide bonds and joining chain. IgM made during the primary response. The first antibody produced in the response to an infection and effective in forming complexes that activate complement proteins. IgM also serves as a B cell receptors. As you can see in this mismatched blood group, the IgM antibody binds to the antigens of the red blood cells and occurs agglutination. This is type 2 hypersensitive. IgM antibody has 10 antigen binding sites. The next one is IgE, immunoglobulin E. Is made by plasma cells. Structure: it is a monomer found in respiratory tract mucosa, lymphatic tissues, mast cells, basophils, and eosinophils in GIT. It is involved in the allergic reactions and responses to parasitic infections and binds to allergens and triggers histamine release from mast cells. As you can see in this diagram, it is a mast cell. It has its own receptors called Fc epsilon R1 receptor. And then the IgE antibody binds to it receptor and it collects for example, it is a pollen allergens and it gives signals to the mast cells release of chemical like histamine to suppress the allergy. The last one, IgD, immunoglobulin D. The structure: monomer, 2 heavy chains, 2 light chains, made by plasma cells. Function: primarily as receptor on B cells, plays a role in initiating B cell activation, initiates immune response. As you can see here, the plasma cell secretes the IgD monomer and goes back to the B cell and act as a B cell receptor. Location: surface of mature B cells, lymphoid tissues. Next one, immunity. Passive immunity, active immunity. Passive has two: naturally acquired, artificially acquired immunity. In natural, IgG antibody crosses the placenta, IgA antibody by milk. And artificially acquired immunity, in case of snake bite, antivenom immunoglobulin act as against the venom. In active immunity, naturally, if I got infected by a pathogen, then my body naturally created the antibodies through immune system. Artificially, vaccines and booster shots, weak or dead antigen given into the body, and immune system produces the antibodies against specific antigen. Next, the structure of antibody. It has the constant heavy chain, constant light chain, variable light chain, and variable heavy chain. It is the antigen binding sites, and it has disulfide bonds. The variable region differ from one antigen to another because it is depending on the antigen shape. The variable region also changes. It is the complement protein binds here. Next is antigens. Antigen is a molecule that able to generate antibodies in immune system. Antigen refers to AG, antigen generator. Initiate immune response when AG binds to AB. Two types of antigens: foreign antigen, self antigen. Foreign antigen has exogenous antigens, self antigen endogenous antigen. But not all endogenous antigens are self antigens. The self antigens originate within the body and through negative selection of T cells that our immune system does not target self antigens. If the endogenous antigens generate from viruses, bacteria within our body, then immune system attacks. Structure of antigen. The antigen can be made up of proteins, peptides, amino acid chains, and polysaccharides. It is the structure of antigen. The elevated structure of antigen is called epitope. It is the shape or amino acid sequence determines its function. The epitope is the smallest part of antigen recognized by our immune system, binds to antibody. The part of antibody that binds to epitope is called paratope. This paratope is specific for specific antigen epitope. Epitope of protein antigen. It has two types: conformational epitope, linear epitope. Conformational epitope, the 3D structure of epitope defines its functions. Like it has a different conformational structures. It is different from one another. And it has a specific paratope to attach this epitope. In linear epitope, linear amino acid sequence defines its functions. Next, the complete antigen, partial antigen. Complete antigen able to include antibody formation and generate immune response. The partial antigen, also called heptin, unable to induce antibody formation, but it can generate immune response when coupled with carrier protein. Heptin, the partial antigen, is two types: complex heptin, simple heptin. Complex has the large molecular weight and it has the low molecular weight. It is polyvalent, and simple heptin is univalent in nature. Complex heptin has two or more antibody binding sites. Here's the exogenous antigen and endogenous antigen. Exogenous, origin outside the body. Endogenous, inside the body, self antigen or antigen from bacteria and viruses. Here, the antigen presenting cells take up the exogenous antigen and processes them. In endogenous, no take up required for endogenous antigens. APC cells present in T helper cells, and endogenous antigen recognized by cytotoxic cells. MHC2 processing. It is the major histocompatibility complex 2. In this, MHC1 processing. The another type of antigen called superantigens. The superantigens are micro-produced toxins that bypass the specificity of the immune response and activate up to 30% of lymphocytes. Superantigens has the ability to trigger excessive stimulation of human and other mammalian T lymphocytes, massive release of T cell mediators. As you can see in this, the super antigen binds to the T cell, an antigen presenting cell up to 1/5 T cells activated, and T helper cells and cytotoxic cells. Hope you understood. Thank you.